very much, Vincent. And first of all, a big thank you to Myeloma Patients Europe. So it's an honor to give this presentation. And I think after dealing with myeloma patients now more than 15 years, it's not only remains a big part of my work, but also, let's say, all the questions, sometimes uncertainties, but also hope is something that is really close to my heart. And what I will do now in the following 40 minutes, and I'll make sure that I will leave enough time for Q&A, is to go through the latest developments in the treatment of myeloma. And I also highlight some of the newest data from the last EHA and ASCO Congress. And indeed, I have 50 slides, but I will not discuss everything at the same level of detail. So let's start with the next slide, which is a standard slide. So here you see my disclosures. And then just to remind you that basically, 20 years ago, the treatment options were just chemotherapy and steroids. They're still there. We still need them, but to a much lesser extent than in the past. Now, currently, the landscape has been largely taken over by immunomodulatory drugs, proteasome inhibitors, and monoclonal antibodies. But now, on top of that, we also have the T-cell redirecting therapies. Next slide, please. This is a slide that is frequently shown because it's no secret when you hear you have myeloma, or when I'm in front of a patient with a newly diagnosed myeloma, then many patients, they ask, well, can I cure from this disease? And if not, how long can I live with the disease? And I always give the same answer, and I say, we cannot predict the future, but we can do our utmost best to try to change the future. And the classical paradigm of myeloma is treatment is followed by a remission. Then there's a relapse. Remission can be shorter, and then relapses culminate until the disease can become fully refractory. The next slide, please. And this is something a couple of years ago we were still dreaming of. That is, are we able to achieve very long-lasting remissions, between brackets, remissions that mean that the disease will never come back in a significant percentage of patients? And without being naively positive, I'm a strong believer that these days we are curing more and more patients with multiple myeloma. Next slide, please. But of course, then it all starts with, how do you define cure? For acute leukemia, for instance, or some forms of aggressive lymphoma, it's pretty straightforward. You should not relapse within 5 years, and then you're considered to be cured. For myeloma, it's a little bit more complex because we talk about one disease, but still the disease behavior can be very different. Some myelomas can indeed behave like acute leukemias. Others can behave like what we call a low-grade lymphoma, which are diseases that sometimes relapse after 10 or 15 years. So there is what we call functional cure, which is the disease is suppressed, but it's not gone. And then, of course, the disease is completely eradicated. But then, of course, you need highly effective treatments, but also the proper ways to measure that. And since a couple of years, the term MRD, initially, it was abbreviated as minimal residual disease. Now it is more called measurable residual disease. And it's, in other words, to try to find the needle in the haystack. And next slide, please. And that's very important because there is level 1 scientific evidence, which is the highest level of evidence, that MRD negativity is the strongest predictor for a very good long-term outcome in terms of what we call progression-free survival and, of course, global or overall survival. And there is a second reason why MRD is very important, is because with the current regimens, and I'll show you that in a moment, we are able to induce very long-lasting remissions. that can exceed even 10 years. And for regulators, the traditional endpoints like progression-free survival and overall survival can then be reached after too many years for pharmaceutical industry to be interested to do a study. Because, at some point in time, they want to gain back their investment. And if they can only start gaining back that investment after more than 10 years, the patent can be already expired. So that could also, let's say, delay innovation. So, as many of you might know, in April 2024, the Food and Drug Administration has accepted MRD negativity as an endpoint for temporary approval of new treatments. And we hope and we expect that the European Medicine Agency will take a similar decision in the following months. Next slide, please. So now, very practically, the first-line treatment for transplant-eligible newly diagnosed patients was in 2021. You give induction with a regimen like VRD, bortezomib, lenalidomide, dexamethasone, or bortezomib, thalidomide, dexamethasone with daratumumab. 4 to 6 cycles, then stem cell transplantation, and then maintenance with lenalidomide. Now, very, very recently, and that's on the next click, I'll do that in a moment, sorry. This is just to support the data that support that. So it's fine, Vince, sorry. No, it's on it's on a later slide. But these are just the data that on the next slide that support the use of VRD. 2 big studies. Initially, it was 1 study where VRD was given as induction and consolidation and in between the transplantation. Those studies also compared additional cycles of VRD instead of high-dose chemotherapy with stem cell transplant. And it was found that when you skip the transplant, that then the disease will come back more quickly. But why I want to show this slide is that with, let's say, VRD induction, transplantation, VRD consolidation, and lenalidomide maintenance, we expect the disease to be controlled for an average of 5 to 6 years. So that's, let's say, the benchmark to be beaten. And on the next slide, the major game changer then was the introduction of the anti-CD38 monoclonal antibody daratumumab, which is an antibody, as we have many in medicine, but that binds on plasma cells. And it's a strong antibody because it also activates the immune system in several ways. The next slide, please. So then the study started by comparing the standard of care with the standard of care plus daratumumab. And so the initial study for newly diagnosed younger patients was the so-called Cassiopeia study. And yeah, they give all kind of fancy names, and many studies with daratumumab, they are named after Greek mythology. But basically, that study used VTD, bortezomib, thalidomide, dexamethasone as a standard of care, which was at that point in time frequently used. And since lenalidomide has become generic, I think VTD is really has disappeared or should have disappeared. But that study is at the approach 4 cycles of induction, transplantation, 2 cycles of consolidation with VTD compared to VTD plus daratumumab. But this study was initiated just before lenalidomide maintenance was approved. So there was a second randomization between no maintenance or 2 years maintenance with daratumumab. On the next slide, and this is the long-term follow-up of the study, and it is so important that we don't take decisions only on short-term follow-up, even if it shows significant benefit. But we also dare to look at the long-term follow-up. And here, last year, the data were released and published after a median follow-up of around 7 years. And what is shown here, that by adding daratumumab even to VTD, and without proper maintenance, because only half of the patients received a short course of 2 years of daratumumab every 2 months, we see already a median progression-free survival of around 7 years. And it was also, very importantly, a significant benefit in overall survival, knowing that many patients in the VTD arm received the daratumumab-based regimen at relapse. So this also reinforces the optimal, or the importance of an optimal first-line treatment. Next slide, please. And this just to show you that also MRD measurement is included in those studies. And just to give you an example, so at the right slide, so this first bar, those were the patients who received daratumumab, both induction and consolidation and maintenance. And we see here that 3 out of 4 patients have MRD negativity in the bone marrow, which is very clinically meaningful. And the next slide, please. So this is, let's say, the current treatment algorithm. So very recently published, it's a consensus paper from European myeloma experts. And here we see that the induction treatment is not anymore VRD or Dara-VTD, but Dara-VRD or ISA-VRD. And ISA comes from isatuximab, which is the anti-CD38 monoclonal antibody from another manufacturer. 4 to 6 cycles of induction, then the transplant, and patients who received only 4 cycles of induction get 2 additional cycles of the same induction regimen, and then maintenance with lenalidomide or even lenalidomide plus daratumumab. Next slide. And then, of course, what we want to practice is evidence-based medicine. Where does the evidence come from to recommend Dara-VRD as a first-line induction regimen? This comes from the so-called Perseus study, and this is very comparable with this historical Cassiopeia study. 4 cycles of induction and 2 cycles of consolidation, here of either VRD or VRD plus daratumumab. There was no second randomization. Patients in the VRD arm received lenalidomide maintenance according to the label, and patients in the Dara arm received daratumumab on top of the lenalidomide, with the possibility to stop the daratumumab after a median treatment duration of 2 years, on condition that the patients had 2 measurements of MRD negativity with at least 6 months in between. So the next slide, please. And here you see these MRD results, where around 3 years after start of treatment, we see that also 3 out of 4 patients are achieving MRD negativity in the bone marrow. And nearly 2 out of 3 patients have, this is the dark red bar, MRD negativity at one of the highest sensitivity level, which means the absence of 1 clonal plasma cell in a background of 1 million normal cells. And also, as shown on a previous slide, this study also confirms that MRD negativity, irrespective of how it is reached, is the strongest predictor for the best progression-free survival. And on the next slide, we see that the median progression-free survival was far from being reached after a median follow-up of 4 years, because even with VRD alone, we do know that it can be around 5.5 years. But a couple of months ago, these data shown on the right graph were released, and it's basically, and I'm not good at statistics, but it's statistical modeling. And it's a model that is frequently used by the UK regulators, by NICE, so the health regulators or the approval regulators. And based on that model, one could predict that maybe with this approach of Dara-VRD plus Dara-Len maintenance, we could envision a median progression-free survival that exceeds 15 years on average, which would make us believe that a significant percentage of those patients will be cured by this treatment. And whereas for VRD, it was in this model assumed to be around 7 years. Next slide, please. So also to give credits to everybody, we have also the ISA-VRD in the first line recommendation, and this is based on the German GMMG HT7 study. Similar approach, VRD induction compared with VRD plus isatuximab. Instead of 4 cycles induction and 2 cycles consolidation, in this trial, everybody received 6 cycles of induction. But then, after the transplant, in this study, there is a second randomization, lenalidomide versus lenalidomide plus isatuximab. And maybe that's the most important part of the study, to know what is really the impact of this second randomization and adding isatuximab. But it will take still many years before the readout of the second part will be known. Next slide, please. And this is just a similar, let's say, presentation as with the Perseus study. We see on the left the progression-free survival. And importantly, we see that giving 4 drugs of induction results in a much better progression-free survival, which is statistically significant, irrespective of the second randomization. And also, the levels of MRD negativity improve with each treatment step. Next slide, please. So what might bring the future on top of all this for newly diagnosed transplant-eligible patients? Well, very recently, the data from this MIDAS trial were published in the New England Journal of Medicine, which is, let's say, the most prestigious medical journal for clinical, to publish clinical studies. And here, the design was, we give all patients one of the most potent induction regimens. Whether here this is isatuximab, you know, isatuximab is from a French company and this is from a French group. There might be some chauvinism, but irrespective, it's an anti-CD38 monoclonal antibody. But instead of bortezomib, here carfilzomib, the second generation more potent proteasome inhibitor, is given. So it's not ISA-VRD, but ISA-KRD. Immediately 6 cycles for all patients. And then patients are randomized according to the fact, is there still measurable residual disease in their bone marrow, yes or no? If not, then those patients are considered standard risk. If there is MRD, then they are considered high risk. So this has nothing to do with cytogenetics. This is just based on MRD. If patients have standard risk, so no residual MRD, then they receive either another course of 6 cycles of ISA-KRD without a stem cell transplant, or they receive the transplant and 2 cycles of consolidation. Patients high risk based on MRD all received at least 1 transplant, but the control arm is here 1 transplant plus 2 cycles of ISA-KRD, and the experimental arm, the traditional tandem transplantation. So that's arm D. And on the next slide, I want to draw your attention to 2 things. First of all, after 6 cycles of ISA-KRD induction, we see that already 63% of patients are MRD negative. So that's approaching 2 out of 3. And then on the right side, we see that there is no difference between either arm A or B, so that's either another 6 cycles of ISA-KRD or 1 transplant and 2 cycles, or between arm C and D, where it's 1 transplant versus 2 transplants. And so this study will definitely now fuel the debate about the role of stem cell transplantation in the near future, knowing that ISA-KRD is, at least in Europe, not approved for first-line treatment. And what it also shows is that once you are MRD positive, after 6 cycles of a potent regimen like ISA-KRD, it's problematic to get the same level of MRD negativity with just transplant or ISA-KRD on top of that, or only ISA-KRD. So for those patients, we need new and better strategies. Next slide, please. And one of these strategies could be to incorporate at an earlier stage the new T-cell redirecting therapies, like CAR-T or bispecific antibodies, to really get rid of this MRD, which is hard to tackle with more conventional approaches. And also several studies are ongoing. And one of the studies, for instance, is the CARTITUDE-6 study. But since the control arm is here Dara-VRD, it again will take a very long time before we know the progression-free survival. So also here, MRD negativity as a surrogate will be very important. Next slide. So just to mention that it's for young patients, but also for the elderly patients, there is fortunately progress. And now the standard of care for those patients is also quadruplet. Dara-VRD or ISA-VRD, or when patients are frail, and they are frail by definition if they are older than 80 years, only Dara-RD. But of course with no transplant, maybe with a few exceptions in the fit patients between 70 and 75 years of age. Next slide, please. Now I will talk briefly about progression after first-line treatment, about relapse. And then it becomes a little bit more complicated because first of all, we need to take into account which regimens were already given. Were they given until progression? Did they cause specific severe adverse events? What is the biology of the disease? Sometimes the disease can become more aggressive. There can be extramedullary localizations. And then last but not least, how is the patient? And also maybe not at first relapse, but after many, many lines of treatment, what are the expectations of the patient? So also here, the good news is that things have improved dramatically. And whereas like 8 years ago, it was mostly what we call doublets, carfilzomib, dexamethasone, pomalidomide, dexamethasone, even lenalidomide, dexamethasone. Then a few years later, and this is the lower part, everything became what we call triplets, so combinations of 3 drugs with many more options. But also the decision tree becomes more complex. And then on the next slide, you see from these EHA EMN guidelines, this is just a summary of everything that's possible. But what we do not want is that it becomes a labyrinth where you get easily lost, or at the end that everybody's interpreting it to their, let's say, personal preference. So what we take into account is, has the patient already received, for instance, daratumumab? And most importantly, is daratumumab given until progression? Is lenalidomide already given? And mostly yes. Is the disease refractory to lenalidomide? And the same for bortezomib. And then you end with all these different strategic choices, knowing that this is just a summary of everything that is approved, which is not the same that it is also available nor reimbursed in specific countries. Next slide, please. But I will, for the remaining time, just briefly now touch upon some of these, let's say, newer agents. And until a couple of years ago, we called the proteasome inhibitors, the immunomodulatory drugs, as the new agents. Then it was on top of that daratumumab. And now we call, let's say, the more new immune agents as novel agents. So the first agent that I will briefly like to discuss about is belantamab mafodotin, or abbreviated as belamaf. And that is an antibody-drug conjugate, whereas daratumumab and isatuximab, or what is called naked antibodies, this is an antibody that is armored with a payload of chemotherapy. And this is indicated as the red star on the left side of the figure, on the left side of the slide. And what it basically does, it brings this payload just to the plasma cell. and then it's internalized by the cell and it should kill the cell. So it's basically immunotherapy, yes, but here the antibody is just a carrier of the chemotherapy. And then on the next slide, I show you the most recent studies with belantamab mafodotin. And some of you might know that this drug was introduced a couple of years ago as a single agent. And it was also approved for relapsed refractory myeloma, but it was then taken off the market. And not because of the toxicities, but just because there was a comparative study with pomalidomide and dexamethasone. And that study didn't reach the primary endpoint. And because of that, the temporary approval was not prolonged. But I would call belantamab mafodotin a comeback kit because they have very strong data in combination. And this is the so-called DREAM7 study, where belantamab in combination with bortezomib and dexamethasone was compared with daratumumab plus bortezomib and dexamethasone. And we see that with daratumumab, bortezomib, dexamethasone, the median progression-free survival is 1 year, where it's 3 years with belantamab mafodotin. So that would basically imply that because the bortezomib dexamethasone backbone is the same, that belantamab is much more potent even than daratumumab, at least when you combine it with these agents. And on the next slide, a similar study, that is the DREAM8 study, compared belantamab with pomalidomide and dexamethasone compared to bortezomib with pomalidomide dexamethasone. And that regimen, bortezomib, pomalidomide, dexamethasone, we call it PVD, is a frequently used regimen for relapsed myeloma. And also here you see that the median progression-free survival, 12 months for the control arm, the PVD arm, and 32 months for the Bela-PD arm. And so very recently, the EMA has also approved Bela-VD and Bela-PD for relapsed myeloma. And now we will see in the near future in which countries it will become available and reimbursed, and how often it will be used, because there are some competitors also on their way of approval or recently approved. And next slide, please. Yes, and before discussing that, belamaf has a specific adverse event, which is ocular toxicity. And of course, that's a little bit scaring when you as a patient, you hear, well, you can have some eye damage. And there's been a lot of talk about this just because it's a side effect that is not associated with any other myeloma treatment. So also for us physicians, it was pretty new. But basically what happens is it causes keratitis. So it causes irritation of the corneal layer just because this toxic payload is taken up also by the cells of the cornea. And this can cause eye irritation and also temporary blurred vision. But it's a compound that has a very long half-life, so that means that it can be given once you achieve a response. It can be given every 4, every 6, or even every 8 or 12 weeks. And these eye toxicities are also fully reversible. So it can be a reason to discontinue treatment, but it is very rare. But of course, let's say in patients who have already eye problems, it can be, let's say, not the ideal treatment. Next slide, please. So then, of course, we come to these newer T-cell redirecting therapies, as we call them. And I think they are mentioned on the next slide. Here you see how they act. And basically, the first thing is you have to have a good target. And for instance, for daratumumab and isatuximab, it is an antigen that we call CD38, which is largely expressed on plasma cells. But a couple of years ago, new and other specific targets were identified, like BCMA, which is called B-cell maturation antigen, which is nearly exclusively expressed on plasma cells and even higher on myeloma cells, and others like what is called GPRC5D. And so these are the targets. But then, of course, you need to have the right tools to target these antigens. And then there is, first of all, on the left side, the chimeric antigen receptor cell, or the CAR T cells, or the bispecific antibodies, which are antibodies that with one arm they bind to this antigen target like BCMA or GPRC5D, and with the other arm they bind to an antigen on a T cell. So these create a kind of immunological kiss between the T cell and the myeloma cell, which has a high chance of killing the myeloma cell. Next slide, please. CAR T cells, let's say, are considered to be the gold standard. But first of all, it is complex manufacturing because you produce a product for an individual patient. And over the past years, we have seen that even big pharma companies struggled to fulfill the increasing demand. But now, and I think they deserve really credits for that, they have really upskilled their production capacities around the globe. And on average, it takes around a month to get the CAR T cells. We call it the vein to vein time. That means between the apheresis and, let's say, the arrival of the CAR T product. But there is a kind of variation on that, and occasionally you have a patient where the procedure needs to be redone. Do we need CAR T cells, let's say, off the shelf or within 1 week? I think it's something we can discuss, but I'm not fully convinced about that. But it's very complex, and it's also very expensive. On the next slide, please. These are the data that currently support the approval of the two BCMA targeting CAR T cells, ID-cell and cilta-cel. ID-cell is approved by EMA and FDA after 2 or more prior lines of therapy based on the KarMMa-3 trial, and cilta-cel after at least 1 prior line of therapy for patients who are also lenalidomide refractory, based on the CARTITUDE-4 study. And those studies compared the CAR T product, 1 single infusion, with prolonged continuous treatment with drug-based regimens. Next slide, please. And then at the last ASCO and EHA meeting, I think very encouraging data were presented. And these are the data from the CARTITUDE-1 study. And that was basically the first study with cilta-cel in relapsed refractory myeloma. Patients had received a median of 6 prior lines of therapy. The large majority of patients were refractory to lenalidomide, to an anti-CD38, to a proteasome inhibitor. These were nearly all end-stage myeloma patients. Those patients who were enrolled in the trial and where patients, their disease could be controlled during the manufacturing process during these 4 to 8 weeks, which at that stage is very critical because the disease is refractory nearly to everything, and you want to control it while the CAR Ts are manufactured. So unfortunately, already 17 patients died during this early course of the disease, and or the early course of the treatment. But now we have the follow-up of 5 years, and there I think we see something that was until recently unthinkable. 1 out of 3 patients who at that point in time had a projected survival of only a few months are still alive and without progression after 5 years. And the majority of those patients were in complete remission and MRD negative. So we hope that a significant percentage of those patients were cured. And I think the presenting author made there a right statement, although I do not want to sound harsh, but he said this is really from hospice to cure, because at that point in time, I think the patients who could be enrolled in this trial were very, very lucky ones. What we also learned from this small cohort is that it's important to have a good control of the disease before the CAR-T treatment. CAR-T is more to be seen as a consolidation treatment rather than trying to quench a fire that is maximally burning. And so patients who have extensive tumor load before the CAR-Ts are infused have fewer chances of a very long-term remission than those where the disease is not gone, but it's more controlled before the CAR-Ts are infused. Next slide, please. And I don't want, let's say, to put one product over another, but we have to admit that those who were first, the manufacturer of ID-Cell, there we don't really see a plateau in the studies. And this could be related to the fact that cilta-cel, for instance, is a more potent CAR-T because it binds at 2 different places to BCMA, and it's also another type of immune cells, and that's learning by experience. And currently other companies or academic centers are even trying to make the CAR-Ts even more potent or more fit. Next slide, please. Because I'll skip that one. So this just shows that the overall survival looks very good with the patients who received cilta-cel, although one could criticize that patients in a control arm, if they can get cilta-cel at the next line of therapy, maybe, and hopefully their survival will be as good. Next slide, please. And this shows that when we talk about high-risk myeloma, we frequently refer to cytogenetic features, that patients with high-risk cytogenetic features also benefit more or less to the same level of extent from CAR-T than those with standard risk cytogenetics. Next slide, please. But when we compare this CARTITUDE-4 and CARTITUDE-1, even with the excellent long-term results of CARTITUDE-1, we believe that giving the CAR-T cells earlier in the treatment journey, that means at first or second relapse, is of much greater benefit than waiting until the disease has become refractory to all the conventional agents. Next slide, please. And on top of that, it's also important that the immune cells are as fit as possible. And if the immune system has been exposed for years and years to all kinds of different agents, we know that some of the immune cells are exhausted. And trying to make CAR-T cells out of exhausted T cells can be a less successful story than using fitter immune cells. Next slide, please. And then very finally, very briefly, about the bispecific antibodies. So targeting these different agents. So currently we also have very potent bispecific antibodies targeting BCMA and GPRC5D. Next slide, please. So just to give you an example, with teclistamab, the number of patients who achieve a complete response or better is lower compared to, for instance, cilta-cel. But those patients who achieve a complete remission and those who are MRD negative, they also have an excellent long-term outcome with bispecific antibodies. But the difference is also that it's a chronic treatment. Next slide, please. Now we have, for BCMA, we have teclistamab, we have elranatamab, and very recently also linfoseltamab was approved, which has, let's say, a more patient-friendly dosing regimen. Next slide, please. And then, of course, no treatment without adverse events. And the T-cell redirecting therapies, and I think many of you know that, can be associated with CRS. ICANS is very rare, but there are frequently cytopenias. We also suppress the normal plasma cells, so low levels of healthy gamma globulins is very frequent. there are more infections, and with some agents like talquetamab, there can be also side effects on other tissues. Next slide, please. And this just is basically a reiteration of the fact that the immune system can be weakened in myeloma due to several reasons, not only the treatment, but also the disease itself. And with each treatment line, we risk that the immune system becomes further weakened. And we need to take measures for that. Next slide, please. And one of the ways of doing that is to follow guidelines for us physicians. And it's also important that patients are aware of that and they can follow that. And I think, for instance, vaccination has been largely under-recognized, that patients should be optimally vaccinated. Prevention is the best way of doing medicine. And if infections occur, we need to start on time proper antibiotics. And then, of course, with the T-cell redirecting agents, the majority of patients will require monthly administration of IV immunoglobulins. Next slide, please. And then with this GPRC5D targeting bispecific antibody talquetamab, we see activities to the same level of extent as the BCMA bispecifics, but there are also some what is called these off-target of tumor on target adverse events. So very practically, the antigen is also expressed, for instance, on parts of the tongue, and the majority of people will lose their taste, or their taste will decrease significantly during the initial phases of treatment. Patients can have temporary swallowing disorders. There can be temporary redness of the skin or exfoliation of, for instance, the hand palms. There is also, let's say, changes in the nails, which is all not, let's say, life-threatening, but it can temporarily impact the quality of life of patients. Next slide, please. And so this is, let's say, another, glimpse of the future, where at the last EHA and ASCO meeting, we saw basically the first results of the tri-specific antibody. So you see biotech remains innovative all the time, which are antibodies that target two different antigens on the myeloma cell, so BCMA and GPRC5D, and then the CD3 on the T cell. And these are very immature data, but they look very promising. And so it could be the best of both worlds, these tri-specific antibodies, but it will take still many years before they will be approved. Next slide, please. And I think that's already my last and conclusion slide. So the good news is that the long-term outcome for myeloma patients is rapidly and impressively improving due to the therapeutic innovation. But still, we cannot say that one size fits all. There is need for high individualized strategic choice. And this is, for instance, based on, let's say, the patient characteristics, but also on the disease characteristics and the response to the treatment. But it's no doubt that this upfront treatment with what we call the quadruplets is of benefit for the large majority, and I would say for all young myeloma patients. But the treatment duration, especially like with bispecific antibodies or even with lenalidomide and daratumumab, it's been largely underexplored. We know when we start, but we have limited data about optimal treatment duration. And I also believe that AI-driven models can further support physicians in, let's say, optimizing the treatment strategy for patients if we can fit the models with the maximum of data. And then, of course, all these treatments are very expensive. And what, for myself as a physician, sometimes feels not right that in some countries the access to this innovation remains so limited just because of economic reasons. And that's also something that the myeloma community should put further on the agenda, and of course we cannot impact on the price, but just continue to hammer that nail that access to treatment should not be too much different according to where you are born if you suffer from the same disease. And sorry for being a few minutes late, but thanks for the attention. I'm more than happy to answer remaining questions.